Welcome to Mr. Chalk's Revision Tips. In this video we will look at interaction with T cells, antibody production and general B cell activation. Humoral immunity and cell-mediated immunity are two types of adaptive immunity in which specific immune systems in your body act. So, antibodies are produced by B cells which respond to helper T cells. So, if we go through how this process occurs. So, to start off with, a specific receptor on the cell surface binds tightly to an extracellular macromolecule, so an antigen, that is recognised. So the plasma membrane then will go and take that antigen into the B cell. A transport vesicle containing the antigen receptor complex is then surrounded and taken in through the plasma membrane. A lysosome fuses with the vesicle. The lysosome releases digestive enzymes into the vesicle, which breaks up the antigens into smaller fragments. The antigen fragments are then bound to MHC2 molecules, and then these will go and be presented to B cells. So, when the B cell encounters a T cell that is presenting the correct shape antigen to activate it, the receptors on the surface of the B cells bind to that antigen. T cell receptors can only recognise an antigen when it's complexed with a major histocompatibility complex on its surface, so that's why it goes and it binds to that. So the T cell receptor binding will go and have other things, so things like glycoproteins will go and bind to other specific fragments of the polypeptide chain that it's presenting. And the helper T cells, in response to this, release interleukins which cause B cells to undergo clonal expansion. So you get lots and lots of B cells all being produced at once. The B cell receptors trigger cell signaling molecules to be released inside of the cell. These cell signaling molecules can often be transcription factors. Transcription factors are then triggered, which cause the gene for the particular antibody to be transcribed inside of the cell. This causes the DNA in the gene to unzip, exposing the gene. So one strand in the DNA is being used as a template. Complementary bases will go and attach to it and go and make messenger RNA. So this in turn leads to the production of antibodies. So the messenger RNA attaches to a ribosome, and the ribosome reads that messenger RNA. The ribosome decodes the messenger RNA in groups of 3. So the groups of 3, remember, are called codons, and complementary transport RNAs will go and bind to each of those codons. The transport RNA is specific to an amino acid that it will go and collect before returning to the messenger RNA. The amino acids are now lined up in a very specific order. So bonds or peptide bonds will form between the amino acids, forming a polypeptide chain. This polypeptide chain will then go and fold up into a specific shape, forming the antibody. The antibody will have receptors that are the same shape as a receptor on the cell surface of the B cell, so will bind to that original antigen. So, some of the things that you could be asked are to name and describe the structure and where polypeptide chains of the enzymes are synthesized, explain the function of codons, and suggest a variety of different protein structures that can be formed from polypeptides synthesized using messenger RNA molecules. So you might want to go pause the video and go and have a think about how you would answer each of those questions. If we go and look at the release of antibodies, the antibodies are now parcelled up into vesicles. The vesicles is transported to the surface of the B cell. The vesicle will go and fuse with that outer membrane and release the antibodies by the process of exocytosis. So, if you were asked something like to explain the role of vesicles in the transportation of materials, these are the kind of things that you might want to go and include. So I would suggest pausing the video now and going and constructing that into an answer to the question above. So if we're going to go then and run through the process again. So macrophages will interact with T helper cells, which causes them to be activated. The T helper cells stimulate specific B cells. Only one will be useful, and that is the one that will make antibodies against the antigen causing the infection. Interleukin 2 is produced by the T helper cell, which stimulates the B cells. Once the proper B cell is found, it is stimulated and divides into many exact copies of itself called clones. So this is the process of clonal expansion. Each cloned B cell then undergoes a maturing process and becomes a plasma cell. The plasma cells are specialised to produce lots of protein, in this case lots of antibodies. Plasma cells produce lots of antibodies. Each antibody is a protein formed in a very specific shape to bind to an antigen. The shape of the binding site is the same as the shape of the receptor on the surface of the B cell. These antibodies are then secreted in large amounts. Once the infection has been dealt with, many of these cells will go and die off. However, some of them will remain and become memory B cells, which form part of the body's long-term immune system. Thanks for watching.